Actimism makes it easier, and we're moving right along with the reaction lab, and we're using yeast. Okay? Now, you should be seeing a video, you should be watching or viewing a video right now with the lab going on. And we actually were able to take this shot inside of an actual lab. And instead of just using sugar, because what happened is that I forgot to tell the kids to bring sugar, and so we decided to change it and just use juice. And because juice has sugar in it, it worked perfectly well. So we mixed half and half, which was we mixed hot water with some fruit juice, and then we mixed cold water with some of the fruit juice. So 50 ml of water in each cup. One was hot and one was cold. So the hot water was put into the cup A and cold water was put into cup B. And then we put 50 ml of the juice in cup A and 50 ml in cup B. So it was like a 1-to-1 ratio. And then what we did, we took packets of yeast. We took 1 packet of yeast, but we were from the same company. We took 1 packet of yeast and put it into 1 cup and into the other cup. So each cup had a packet of yeast. Over time, we left it there so that the reaction could go forth and we could see the difference in reactions based on temperature alone. And then, upon doing that, based on observation, I would have asked my students to write up a lab. So, here it is. We have the lab, and right about now, I'll be explaining the lab, and you'll be watching it. You'll be observing as well, which is pretty cool. That's power of tech. So here we have name of members in group. If you didn't work in a group from my thing, because you could either work in groups in terms of if this is a second farm lab, I will let them work in groups. But seeing as how some people could do this as a CXC lab as well as just the first part, like leading into real labs, I would tell children, well, let's do this as a lab because, so they could get reintroduced to the topic. Because sometimes you need a little help, you need a little push. So it says here name of members in group, and so I'm going to help, right now I'm going to help my kids, to write up the lab. That's the whole purpose of me doing these videos, to help you and to also help my children that I teach and my students as well, because they're older ones, to learn how to write up a lab properly. And so we have all the basic parts of a lab. We have the name, the date, SBA topic, title, aim, materials and apparatus, method and all that. So this would have been sent to the kids right down to the table until they too can build their own table. So all this is a learning process. Now, I would have gone ahead and helped them somewhat. And it starts off with my name, Miss Blondell, and the date, which was, we did this on the 6th of October. So today I am going to change the date to the 8th, which is today, and SBA number 2. Well, let me keep the date because then we did it on the 6th, so I'm just going to help you out now. So this is the second SBA, because the first SBA that they did was, what did they do again? Oh, osmosis. So we did the first SBA together, which was the potato and the sugar and all that. And so now we're doing this one. So rates of reaction, we have that. Exploring the effects of temperature on yeast. And then the aim is to investigate how temperature affects the rate of reaction on yeast. And then we have the materials and apparatus. Now, what we had to do, we had to adjust this section because remember we didn't have the sugar. So we adjusted this to the 50 ml of hot water mixed with 50 ml of fruit juice and 50 ml of cold water mixed with 50 ml of fruit juice. We had 2 beakers and 2 packets of yeast and a ruler. Now the method started off, I started off with writing it. So here we have approximately 50 ml of warm water was added to beaker A, while approximately 50 ml of cold water was added to beaker B. Approximately 50 ml of fruit juice was added and dissolved in each of the beakers. And then down here in red, the rest of the instructions. This is like an example for the class, and I would have typed it up with them. And just like that, I'm going to type up this one with you as well. So here we have now, the next one is number 3, and it says, add 1 packet of yeast to each of the beakers. So it's in red right now, but I'm going to turn it to black soon. So you'll say 1 packet of yeast was added to each of the beakers. Good. And now we could highlight this and make it black. Make it black. And we could get rid of number 3. Now number 4. Observe both reaction mixtures for approximately 10 to 20 minutes. So we say both reaction mixtures were observed for approximately 10 to 20 minutes. We could delete number 4 right now, and then change 5. So number 5 would be the heights of the reaction mixtures were measured. And then number 6, so we could delete number 5 now. Number 6 would be, it says record all observational results in an appropriate table. So we say 6, all observations and results were recorded in an appropriate table. Okay, and we are done. Great. So what I'm going to do, I'm going to highlight this in black now, and that is our method. This is what was done. Then I'm going to leave this blank so that when it's printed out, I would just draw it on, and then the diagram, and then that will be the diagram showing how lab was executed. Then moving along, so this is, so let me make it big for you. So this is how it looks. We have everything, and again, I would always urge you to type up your lab, and I hope your lab teachers are allowing you to do this, because it saves so much time. It's quicker. It's just moving along with the times. Let's move on, and so it's more efficient. That's the key word, efficiency. And it's about working smart and not too hard, because why would you have to build a house with bricks when you could just use a bulldozer, right? So moving on, before I get into my long speech, here we have the observations that's pre-written. So the yeast reacted faster in the warm sugar solution than in the cold sugar solution. And then we have the data and results. We have the table, the title. Table 1 showing the data obtained from the lab. And then we have the condition and the height of the yeast in the reaction mixture. And this is what we got from taping. We taped it, we took it for, we let it sit for 15 minutes, and this is what we obtained. And because the centimeters is up here, we don't have to write 8 centimeters and 3 centimeters, right? So for discussion now, for discussion we had some questions. So let me go back to the, let me go back to the first sheets. And here we have the guided questions where it asks you, what is yeast? And in paragraph 2 is, how does it behave with sugar? And then B, in the same paragraph, say, why did the yeast behave differently in the warm sugar water solution as opposed to the cold sugar water solution? So let me teach you how to do that. So some teachers won't give you guided questions, but I do, because I think you need a little help in the beginning to get the show on the road, and then eventually you're going to catch on to it. That in a discussion, you always try to start off with the you start off with a definition of whatever you were studying. Like in this case, you're dealing with yeast, and then you could also include a little rate of reaction. So how you do that? Go online. You type in yeast. Open up. You type in yeast. Oh boy. Type in yeast. So you go into, well, more than likely Google or Bing, whichever one you have, and you type in yeast. And no, I don't want to study the yeast infection. So I just want yeast, saying yeast infection, but that's not the one you want. You want what is yeast. So let's say, what is yeast? And you go. And it says yeast is a microscopic function consisting of single oval cells that reproduce by budding and are capable of converting sugar into alcohol and carbon dioxide. And that's pretty much what yeast is. So you could highlight it and actual copy, and you could place it right there as a definition. My mouse being funny today. So here we have it. So you can't write a microscopic, but you have to say what you're talking about, right? So you say yeast is a microscopic fungus consisting of single oval cells that reproduce by budding and are capable of converting sugar into alcohol and carbon dioxide. Of course, you could change up something if you want to. You could change it up if you want to, if that's your thing. But I'm just doing this quickly. And try to change it up as much as possible. Like for instance, this yeast is a microscopic fungus consisting of single oval cells. I don't even know how to say it. Produced by budding and are capable of converting sugar into alcohol and carbon dioxide. Right, so that's pretty much when yeast is added to sugar. Okay, so then we could end it right there. Budding. And we could send on the next one. When yeast is added to sugar, sugary solutions, it produces alcohol and carbon dioxide. Right? Yeast, the reaction mixture in cup A rose higher than that in cup B because the yeast cells had more thermal energy to react. The more energy something has, the faster it reacts. Okay, so when yeast is added to sugary solution, it produces alcohol and carbon dioxide. The reaction mixture in cup A was higher than that in cup B because the yeast cells had more thermal energy to react. The more energy something has, the greater is its rate. The more energy something has, is its rate of reaction. Right. The more likely particles are able to surpass the activation energy threshold. Right? Great. So that's that. So yeast is a microscopic fungus consisting of single oval cells that reproduce by budding. When yeast is added to sugary solutions, it produces alcohol and carbon dioxide. The reaction mixture in cup A rose higher than that in cup B because the yeast cells had more thermal energy to react. The more energy something has, the greater is its rate of reaction. The more likely particles are able to surpass activation energy threshold. Alright, and the limitation now. One limitation could be the age of the yeast or the conditions of the environment within the lab. Right, because if it's too cold in there, then if you don't have it in a warm water bath to keep it warm all the time at the same temperature, then more than likely you're going to have a problem. It's going to cool down and it's going to slow down the rate of reaction. Now precautions, one example is the beakers are handled with care, and here are others. Water was poured into the beakers carefully, hot water was handled with care. Now a reflection is such that you give a brief but exciting excerpt on how this lab relates to real life. And for example, yeast is used as a raising agent in bread making. Seasoned bakers knead dough a day before to allow it to sit overnight in order for it to rise. Once the dough is processed in this way, the bread produced will be light and fluffy. Right? And so conclusion, the higher the temperature, the faster the yeast cells will react when placed in a sugar solution. But do note, this could be misleading because if the temperature is way too high, then the yeast cells will die. They could only work at optimum temperatures of between 32 and maybe 37, between 32 and 37. Anything higher will destroy the yeast. They'll just die. They'll be too hot. Well, I hope this helps, and I hope that you get through. Keep on pushing, keep on thriving. See you, I'm out. Oh, by the way, did you see the stuff rise? I hope you did. I hope you enjoyed the lab as much as we did. Good night.